Hello everyone. After a long gap, I'm making this video. And really sorry, I'm not keeping right now good health, but still, there was a lot of requests from the students, and yes, I should keep on going with that topic because somewhere I've started something. I should end it also. So now moving on with principles of inheritance. This is part 2. So we in the previous section we have dealt with laws of Mendel and we have seen monohybrid cross. Now let's move on and see that there are certain examples which uh, how we will explain it and which shows deviations from your Mendel's law. Now this is your what? This is your incomplete dominance. Now if you take this case, here it is the case of Snapdragon or Mirabilis japa. This is given in your NCRT book. Okay, so from there only I have taken it, and in this diagram, if you look at, if your red plant is crossed with the white plant, okay, and then what do we see? The gametes will be formed, capital R and small r, and now you know how to form the gametes. Now when these two gametes fuse together, they form the zygote and F1 generation. Instead of Mendel's, according to Mendel's law, here we should get what? Here we should get this genotype. So according to the Mendel's law, it should be red. But what we see here is, it is saying that it is all pink. It is not red. So means, and now after self-fertilizing the this F1 generation, you know, self-fertilization is done. On self-fertilization, you see, you will get both the gametes at this end. This is the gametes which is showing, and now you can just multiply it. I hope by now you know how to form the individuals. So just unite the gametes, you will get the individuals. So here we are getting, and we are getting phenotypic ratio, what? Red, pink and white in the ratio of 1 is to 2 is to 1, and the genotypic ratio is also what? That is 1 is to 2 is to 1. If you remember Mendel's monohybrid cross, the phenotypic ratio was what? It was 3 is to 1. It was 3 is to 1, but in this case it is 1 is to 2 is to 1. Now how to explain it? How to go about this? Okay, so see, very nicely it has been explained. You can see here that the modified allele, okay, the modified allele could be responsible for the production of, like in this case, the modified allele. Let's see what is the modified allele and what it does, and then we will conclude which is the modified allele, whether it is capital R or small r. Okay, the modified allele could be responsible for the production of a normal, a less efficient enzyme. It should, or it would be possible that it is leading to the functional, non-functional enzyme at all, or no enzyme at all. The phenotypic trait will only be dependent on the functioning of the unmodified allele. So in this case, if you look at, your unmodified allele is what? R, and your modified allele is your small r. And now since here you can see here the unmodified allele, which is representing the dominant allele. Okay, the dominant allele is your unmodified allele, and the recessive allele is your modified allele. This is modified allele and this is unmodified. Now here, why the color is pink? Now what is happening here in this case, because this small r is not producing any enzyme at all. So what is happening? This color is due to pigmentation. Okay. Now the more the amount of the enzyme is produced, the more the pigmentation will be there. As I hope you know that there are different kind of reactions in our body. Okay, and one enzyme is responsible for, suppose, carrying. Suppose I carry write a reaction. Okay, suppose x1 2 and it forms here, suppose x2, then it forms x3, type of product. Now for this, the enzyme 1 is required and enzyme 2 is required. Okay. Now the R is forming the enzyme. Okay, but small r is not forming any enzyme. So the amount that is formed here of the pigment is, in this case, is less in compared to this kind of genotype where it has got two alleles of the same type, and therefore more number of enzyme production is there, and therefore more formation of pigment is there. Okay, so because of this, it is leading to a kind of a condition which we are calling as incomplete dominance. Okay, so uh, this is one of the classical example, and remember it. And here the genotypic ratio and phenotypic ratio is same. Okay, now moving. So we are now moving on to codominance. So codominance is what? It's a case where your no there are. It's also a case of blood group is a very good example where it is a case of codominance, means both the alleles are able to express themselves. Okay, so we call it as codominance, and it is also a classical example of multiple allelism, means that many alleles are responsible for that particular character. Not only two alleles are there for that character, but it has more than two alleles. Okay, that is what we call as multiple allelism. But remember one thing, that multiple allelism can be seen in a population. Okay, it can be seen in a population but not in an individual. Okay, we can see it in a population but not in an individual. In an individual, you can see only the two type of alleles. Why? Because you have got only a pair of homologue. So on this homologue, you can have now on this homologue you can have allele, and this homologue you can have. Okay, not more than that, isn't it? So there can be only two alleles in an individual, but a population can show many alleles. Okay, now this blood group is regulated by three kind of alleles. What are they? They are your IA, IB, and your small I. Okay, now if I have a blood group A, now this blood group A can be represented by two type of alleles, allelic composition, that is your IA, IA, okay, or IA small I. But if it is blood group B, it can be represented by IA, B, IB, IB, IB and small I. But if it is AB, it is represented only by one allelic composition, that is your IA, IB. No other allelic composition is there, and this is the case. This is the case of your codominance, where both the dominant alleles are able to express themselves. And blood group O will have only one type of allelic composition, that is both the recessive alleles. Now in a population we are finding all kind of individuals and these three kind of alleles. Okay, and in class 11th you have read one thing. So you can see here that this blood group A has got antigen A and antibody B. Okay, and B blood group has got antigen B. Remember, what is antigen? It's a glycoprotein coat over your RBC. Okay, now what do you see here? That antigen A is present in blood group A and antibody B is present. Blood group B has got antigen B and antibody A. AB has got both the antigen, no antibodies. And here no antigen, both the antibodies. And that's what we see here. Since it has antigen A, therefore it is reacting with antibody A and clumping. But it has no antigen B, therefore it is not reacting with antibody B. When artificially we are providing them. Blood test जब करते हैं, तो this is how we know the blood test. We do the blood test. Okay, so if antigen B is there, here, so you can see that it is reacting with antibody B. If it is reacting with antibody B, your blood sample, then it is your blood group is what? B. If it is reacting with both, then your blood group is what? AB. If it is not reacting with both the antibodies, then it is of your blood group is what? O. That's how we come to know about the blood group. And even if we know the blood group of a couple, we can predict. Now, suppose I'm taking you an example. Suppose the couple, the mother is having blood group A, and the child is having, and the father is having blood group B. Then I can have here the progenies of both, all the kind of blood groups. You can see here, you can make it from here, IA, IB. Okay, then you can have progeny here, which is what? IB. Then you can have progeny here, IA. Once we have done that, okay, again I will do. Then I can have progeny like this, IAI, and then small I, small I. ज़्यादा बन गया, ये 2 बार बन गया आपका, ठीक है. तो you will get all kind of individuals in this kind of genetic composition. To some extent we can tell, if we know even of the child and the mother or a father, then we can predict to some extent about their genotype. Okay, and remember this about blood group. Okay, and that's why. Okay, now we are moving on. There was something clicked me, but it's okay. Okay, now moving on to the hybrid cross. Okay, that is your inheritance of 2 genes. Okay, we have seen monohybrid cross, that is inheritance of a single gene. Di means 2, hybrid, so we are going to see inheritance of 2 genes. Okay, now this was Mendel's experiment. Again, coming back to the Mendel's experiment, what do we see here? This is your F1 generation. Now here round and yellow seed here too. So this is the dominant character, and wrinkled and green he too. And when F1 generation, what did he get? He get all round and yellow. Then what he did? He allowed to do selfing. He did selfing, as you can see here, and then he got, you can prepare these gametes. I hope you know how to prepare the gametes by now. So let's make once more for you. I am doing it here. Why? Now, how will you make the gametes? Since they must be present on your homologous chromosomes, so homologous chromosomes will separate. So R will come on one side, both the R's, and you will get both the Y's, the Y and Y. And another composition could be your capital R with the small y, okay, and small r, small y. So this is how the 4 combination of gametes you are going to get. Okay, now this is what is shown here in the diagram, as you can see here. So all these are your gametes, which are now uniting the gametes. On uniting the gametes, what do we get? Here we get what? Round and yellow in 9 ratio, and what we get? Round and green in the ratio of 3, wrinkled and yellow in the ratio of 3, wrinkled and green in the ratio of 1. If you look at carefully, this and this are your parental generation, but round and green, wrinkled and yellow, which are your what? They are your new combinations. They are your recombinants, which were not existing in the parental generation. So they are called as recombinants, and they are occurring due to assortment of your chromosomes. Okay, so chromosomes are shuffling, and because your what is happening? Had your I, capital R, always goes with Y, and small r always goes with small y, then you won't have got these recombinants. You got them because they assorted independently, because capital R went independently of the other non-allelic gene. These are 2 alleles, these are 2 alleles of a gene, and R and Y are what? They are non-allelic. They are non-allelic genes, and these non-allelic genes, they segregate independently of each other. This is what is independent assortment. So what does independent assortment say? Let's look at it. The Mendel then generalized the statement based upon his hybrid cross, and he called this law of independent assortment. He said that when the two pair of traits are combined, he said when the two pair of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of characters. Okay, so this is called as and I have shown it here on a chromosomes also. Let's look at that diagram, because that from here only your Sutton and Bovary came and they gave your what? The law of chromosomal inheritance. They said that the genes are present where? On the chromosome. They gave this chromosomal theory of inheritance, isn't it? Now, if you put them, these genes on the chromosome, how the picture would look like, I'm trying to show here so that you get a view. Okay, how they are placed on the homologous chromosomes and how these homologous chromosomes segregate during gamete formation, you know that. And since they are assorting independently of which, how you are getting now 4 different set of gametes on a chromosome. That's what I've shown here. So you can see here, this is one homologue, this is one homologue, and this is another homologue. I hope it's clear. It is showing BB and it is showing SS. One capital S and this is small s. Now what is happening here in this diagram? I have separated like this two and this two. Okay, so homologous chromosomes have got separated, and this one homologue here and one another homologue here, and that's how you got this combination. Okay, and that's how you got this combination. And now when the homologues, I just interchanged it and then separated them, and that's how we got this two combinations. Okay, so this is the basis of your, that's what was said by Sutton and Bovary, that genes occur in pair and chromosomes occur in pair. Okay, and the genes segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. So this one is for your genes. Remember, there is a question in your NCERT is asking, so how will you tell it? If you read the statements very carefully, it will be clear to you. Okay, so what it is saying that during gamete formation such that only one of each pair is transmitted to a gamete, and segregate at gamete formation and only one of each pair is transmitted to a gamete, है ना? तो अगर हम इस पे भी gamete रखेंगे तो हम इसको भी इसी तरह करके देख सकते हैं. Independent pairs segregate independently of each other, and one pair segregates independently of another pair. Okay, so one pair of chromosomes segregate independently of another pair. Okay, and independent pairs segregate independently of each other. Okay, so if you look at now, this is what you can see. Okay, so you can keep here and you can just do it. Okay, so let me put it here. Why it's going off? So suppose I put here your yellow, yellow, and this is for round and this is for wrinkled. Then you can see how the, now first you know that what has happening is that the duplication of chromosomes takes place. So you know that, so they have duplicated, so both will be here what both will be, where is the pen? So here both will be capital Y and here both will be small y. Here both will be capital R and both will be your small r. And now when they will segregate, and then you will get all things you know about it. Okay. So now moving on further. Now if you look at pleotropy. Pleotropy is what? When a single gene, you know, exhibits monophenotypic expressions. Such a gene is called as pleiotropic genes. Like, you know that in your metabolic pathway, one enzyme may be involved in many metabolic pathway, and that enzyme will be contributing to what? You know, will be producing multiple effects. So same thing here, that a single gene is there but producing multiple effects. Okay, so here it's a case of a gene, effect of a gene on metabolic pathway. Okay, so maybe it is forming certain enzyme, and that enzyme has got roles in different reactions, and therefore we are seeing different kind of phenotypic effects. So a classical example in this case is your phenylketonuria. In this case, what is happening in this disease? There is a mutation in the gene that codes for the enzyme phenylalanine hydroxylase. Now you look at it. This is an enzyme, and a single gene mutation is caused here, and this manifests itself through phenotypic expression characterized by mental retardation. Those who are sufferers of it, they are suffering from what? They are suffering from mental retardation, reduction of hair and skin pigmentation. So because of this enzyme, this enzyme is defective enzyme which has been formed in them, that is phenylalanine hydroxylase. Okay, so now this is what happens in this disease that is called as phenylketonuria. In them, because of this lack of this enzyme it converts what? The amino acid phenylalanine into tyrosine. So phenylalanine hydroxylase converts your amino acid phenylalanine into tyrosine. Now since it is not able to do, what is happening? Your phenylalanine is getting accumulated in your body and getting converted to what? Phenylpyruvic acid and other derivatives. Accumulations in the brain result in mental retardation, okay, and also excreted through urine because of poor absorption by kidney. Okay, so that's why we see its multiple effect, and such genes are called as what? Pleiotropic genes. Now the last one which is dealt in your book is your polygenic inheritance. One of the classical example of polygenic, where polygenic means what? Many genes. They are non-allelic. Okay, so remember that these genes are not present at the same loci. They are present at different places, and these genes are non-allelic genes. Okay, there are three different genes which is responsible for skin color in humans. Okay, therefore it is a case of multiple genes. हाँ, पॉलीजीनिक इफेक्ट इसीलिए कहते हैं इसको, पॉलीजीनिक इनहेरिटेंस कहते हैं. एक है मल्टीपल एलिलिज्म which was your blood group, there was a single gene having different alleles. So loci was same in that condition. The location of the gene in the case of multiple allelism was same, but the type of allele was varying. Okay, but in case of polygenic inheritance, the loci is even different and the gene is also different, and it is an additive effect of the genes. Okay, so there are three genes, and their combinations will determine what kind of skin color it will produce. Okay, so it has got an additive effect. Okay, so you can see here human skin color is another classical example, and the effect of each allele is additive. To understand this, let us take three genes A, B, C. है ना? if suppose an individual, now in this diagram it has shown very nicely, you can see here it is showing very dark skin color. Why? Because all of them are what? All are not dominant genes. All are dominant genes. Like they are having like A, B, B and C. Now if it is very light, then here the gene composition is what? It is small a, small b, small c. Okay, now within a population you can have different permutation and combination of these alleles, and what do we find? That majority of the population is falling in within this range. Now very less will have, these are the two extremes, having all the dominant genes and having all the recessive genes. Okay, so most of them in a population, if you look at, they are having a mix of it. That is, they are having a majority of them. If you look at, now it is showing here also, it is showing this kind of gene composition, that is mix of it. Okay, so this is your what kind of curve? This is a bell shaped curve. So polygenic genes, basically whether it is of height or it is of skin color, you will see height is also what? Whenever you see polygenic inheritance, you will see that there is a continuous variation in that. Now all kind of heights you see, all kind of skin colors you see. Now you don't see only this type or this type. Like we saw in case of Mendel's, they were contrasting traits. Here no, they are continuous traits. Okay, they are varying continuously. Okay, so it's that case your skin color is having all the shades of skin color. Okay, so here what do we find? That the additive effect of the genes and polygenic inheritance shows what? Bell shaped curve, because the majority of them will lie within this composition. Okay, where it will be a mix of dominant and recessive allele. So majority of them will lie in this range. Very few will lie at the extremes of both the ends, and that is what you see. So therefore polygenes, whether it is of height or whether it is of your skin color, it shows what? It shows your bell shaped curve. Okay, and still if anybody has any doubt regarding what I have covered, you can text me and you can ask me. Thank you for observing.